So this is a Nature article that is open access, and I found it while I was looking at what are the effects of sucking in particulate matter. What does it do to our body's immune system? What does it do to our lungs? All that good stuff. I popped the link into chat so you guys can look at it. So in this article, first of all, I just want to show you this really awesome picture. These are lymph nodes that have been taken out of dead people, unfortunately. These are at different ages, and these are representative lymph nodes from those people. And what the black stuff that you're looking at are carbon particulates that accumulate in these lymph nodes with age. It's pretty gnarly. Let me just go through the introduction to give a little bit of context about this. So, inhaled particulates from environmental pollutants accumulate in macrophages and lung-associated lymph nodes. So again, I mentioned macrophages before. They're these little cells. They have a lot of like little, almost like little fingers. They look like a skirt, and they like move around, and they like pick up stuff in their environment, and they take it back to this thing called the lymph node. You have lymph nodes all through your body. A fun little diagram that we can look at together so we can quickly see where all your lymph nodes are. Now, what I want you to take a note of is the green lines, like they look almost like blood vessels innervating the body. And those green veins are connected to these little green sacs. And those are the lymph node. They look like little peas. You have lymph nodes, for example, you have some that are like right under your, kind of behind your ear, somewhere around your, like under your, whatever this is, your chin, neck thing. And when you get sick, these lymph nodes get swollen, and they can get really painful when they're swollen. If you like touch them, if you guys are sick right now, you might want to like touch your lymph nodes. They'll feel like a hard ball. And the reason they're big and swollen is because they're filling up with T cells and B cells that have been activated by things like your macrophages and mostly your dendritic cells, which are another very important type of cell. In fact, it's your dendritic cells that are doing a lot of the activation of your T cells. And once your T cells get activated, they turn on this program where they'll proliferate and make clones of themselves. And these clones of T cells are T cells that recognize whatever sort of pathogen the dendritic cell or macrophage presented to it. So they make copies, and then they go out from the lymph node, and they go, they follow these little like chemical trails from the site of the infection, and they follow that gradient. And that's how your T cells go from like a lymph node to wherever the source of your infection is. And that's why, again, because you have this huge expansion.
Dude, how do you feel?
Because you have this expansion, that's why your lymph nodes feel sore. Anywho, that's just a quick little aside. So going back to these lymph nodes. So again, they saw that inhaled particulates from environmental pollutants compromise immune surveillance. So this is how well your body responds to things via direct effects on immune cell function and lymphoid architecture. So this is just talking about how well the lymph node is structured and stuff. So what was their question? The world population is aging, and the majority of healthcare costs, morbidity, or mortality that are associated with disease concern individuals in the 6th decade of life and older. So if you're above 60, you're going to start thinking about your health. That makes sense, although you should be thinking about it now. So let's see. The question was, they noticed that in SARS-CoV-2 pandemic, elderly individuals have an increased susceptibility to respiratory infections, as well as other lung diseases, such as chronic obstructive pulmonary disease, or COPD, and cancer. This susceptibility is attributed to senescent changes in the immune cells, which result in systemic inflammation and functional impairments. What happens when you age is that there's this organ in your body called the thymus that makes that basically that's where your T cells mature, and it shrinks. Well, the useful part of your thymus shrinks because it's getting filled with fat. So if you look at a young person's thymus and an elderly person's thymus, you're going to see a lot more fatty deposits clogging up that thymus, and it's not going to be churning out the mature T cells as quickly as a young person's thymus. So this is already a strike against your immune system. And there's many other changes that happen in an aging body, not just environmental, that impair your immune system and its ability to respond to things. Now, this paper, though, is looking at what about environmental impacts? Does that change how well your immune system can respond to insults? So they were looking at human tissue samples. They took these samples, they were from dead people, human organ donors, and we consistently observed that lymph nodes associated with the lungs, so the ones around here somewhere, were black in color owing to the presence of black particulate matter, whereas gut-associated lymph nodes, for example, were the expected beige color with no particulates. Thus, we began to investigate the effect of particulates on immune cells and lymph node architecture in these different lymph node sites using quantitative imaging and cell and functional assays. We found that the atmospheric particulate matter accumulated with age, specifically in lung-associated lymph nodes, so the ones in your lungs, but not in gut-associated lymph nodes. I'm not sure why they choose to keep going back to gut-associated lymph nodes, because you do have many other lymph nodes as well, like even in your leg and like, yes, in the stomach area, to see if your lymph nodes there are swollen. I don't remember the name of those lymph nodes because I'm like really bad with anatomy. So I don't remember what those ones are called off the top of my head. But anyways, it's interesting. They are just comparing everything to the gut lymph nodes. So they noticed that the accumulation of this black particulate increased remarkably after age 40, which is interesting and slightly weird. They're called undernodes. These are my undernodes. Google cracked down on full-size diagrams and it sucks now? Really? Wait, this is a real thing? Why? I think you can buy thymus extract on Amazon. I'm not sure if it would help regenerate. What? Special? Hey, so special. Welcome to stream. You're telling me that you can buy a thymus extract on Amazon. I've got to see this. Hang on. I'm going to open this in a different browser because I don't want to accidentally dox myself. Thymus extract. Oh God. It's a thing. Wait, what? It's on WebMD? This cannot be right. This is what WebMD says. The human thymus gland is responsible for the production of some important immune cells, such as T cells. Thymus extract is produced from the thymus glands of cows. Some people use it as medicine, but the quality and potency of thymus extract products can vary greatly. How does it work? Thymus extract may work by improving or boosting the immune system. Thymus, what? That's all they have about it. Oh, they have more things. Early research shows, okay, let's see what it's possibly effective for. Weakened and enlarged heart. Early research suggests that taking a calf thymus extract along with usual therapy improves heart function. So let me back up here and talk about what is a thymus, because this does not make sense to me. This is absolute bullshit. Let's look at a thymus. Let's look here. And because I have it zoomed in, it's gonna look hella weird. Let me go to images. And we're gonna have to just look at one of these really small diagrams. What do I want? This is fine. Okay, you guys can kind of see this, right? This looks pretty clear. So the thymus, again, it's located right above your heart, and it shrinks and gets full of fatty deposits as you age. But when you're very young, your thymus is very big. And it's composed of these epithelial cells, like the sort of matrix that kind of gives your thymus structure. Those are like epithelial cells. These are like structural cells. And they form like this mesh. And and on the surface of these cells, they're presenting like peptides and stuff to train any baby T cells that come into the thymus. Basically, they're like selecting for T cells that don't recognize self-peptides. Why is that important? If you have T cells that recognize your own peptides, like your own proteins and whatnot, that means, and if those cells get released out into your body, they will start attacking your own organs and your own cells. So your body has to make sure that does not happen. So in the thymus, if there's any self-reactive T cells, it kills them. It sends them a death signal. Let's just call it that. Now, any T cells that do not recognize self proteins, those are allowed to pass, and that's a very small number of T cells. Now, again, the thymus is mostly composed of either your epithelial cells that are forming this little matrix for your T cells to kind of hang out and migrate through, or it's also composed of dendritic cells that are also doing the same thing. They're like presenting peptides to your T cells. So the dendritic cells here, they're these little brown things. And then the little blue things, I think these are what, macrophage? Oh, these are like cortical epithelial cells. And if we want to look at like a scanning electron microscope image of the thymus, you'll get to see the matrix a little better. It's really cool. It's really beautiful to me. This is my favorite image.
You think that's air you're breathing now?
Let's close some of these tabs. Oh, it's really small. God damn it. Okay, this is not great. Those green things are your cells. Those green pea looking things, those are your T cells. And then the blue stuff, that's the matrix that makes up the thymus. Now, if you were to like take this out of a living animal and grind it up, you would be killing all of these cells. I don't know what biological or like what compound this thymus extract is supposedly carrying that has whatever health benefit these people are claiming. It makes no sense to me. I think it's bullshit. I'm gonna quickly look once more at what this says. Like, it's not subject to FDA guidelines, so it's not FDA approved, it seems. Yeah, I'm suspicious of anything that says grinding up a thymus and consuming it is going to make you healthier. Wow, sorry, so special. I just really just attacked that point. My bad. Maybe, you know, maybe there is something out there that claims it does stuff, but I've never heard of it. First lymph node I had go bad was in the groin. Ooh. Ow. There's a lot of extracts like this. I know of kidney and liver extracts claiming to do the same for those organs. So the one shred... Oh, hi, Pandarius. Welcome in. The one sort of possibility I see with extracts maybe having some effect is that there are growth factors and other sort of like bioactive molecules that are still in their preserved active state, and they can bind to maybe like some growth receptors in a person who maybe has like a bad thymus, and somehow those growth factors could maybe help the organ. Kitty, did I say kitty extract? No, I did not say kitty extract. But that's just my speculation as to why these extracts could work. I shouldn't diss on it so much. It just seems incredulous to me, but I truly do not have enough background information to maybe totally rip it apart or support it. And the funniest one for me is people eating sea cucumbers because they think they can regrow because sea cucumbers can regrow their organs, thinking it's going to give them the ability to heal their own organs. People do that? That's wild. No, that's not how it works. Okay, going back to our little lymph node story. Right, so we were talking about these lymph nodes here. These are, so at age 20, you should have a beautiful, sexy lymph node just like this. If you're a 20-year-old, your lymph node is lovely. It's delicious. I could eat it for lunch. Did not say that. 29, it's starting to get a little bit more gross. Now, these are lymph nodes where they've actually cut them open. This image is kind of blurry, so we're going to zoom in a bit to see if it gets a little bit more clear. At 29 and 30, it's interesting they took these two time points. Maybe this was just what was available. Again, they're looking at deceased organ donors here. You're starting to see more particulate matter. And as you age, after age 40, you're getting some really gnarly looking lymph nodes. Then they like section these lymph nodes into really thin sections, and then they look at them under a microscope. And so what you're looking at here is just a cutaway of your lung lymph node. So LLN is lung lymph node, and MLN is, I think, mesenteric lymph node. Your mesenteric lymph nodes are located behind your small intestine, I think, somewhere. When I'm trying, so I'm really bad at this, but one thing that we have to do, because I study the immune system, is sometimes I have to take the lymph nodes out of the mice that we use. And the mesenteric lymph node is the hardest one for me to find, because you literally have to pull the entire intestines out, and then there's like this fatty bit of tissue, and in that fatty tissue you like pull it apart like a sheet, and there's this beautiful lymph node that's just kind of hanging out there, and that is the mesenteric lymph node, but it's so hard to find. The easiest lymph node for me to find is the ileal lymph node, which is like located kind of around the leg of the mouse. So going along, we see that the control here is the mesenteric lymph node. They said that they did not see dark particulates in the mesenteric lymph node, which is in the stomach, only in the lung associated lymph nodes. Also, welcome in Roslyn. Thank you so much for that follow. I hope you're doing fantastic today. So look at this. Look at age 73 and then age 56. There's all of these particulates. We'll look at the legend and see what it says. Gross appearance of lung-associated lymph nodes over age. These are bright field confocal images of human lymph nodes, lung lymph nodes, and mesenteric lymph nodes obtained from organ donors of various ages. You know, interestingly, a complaint I had in this paper is that they did not say how much exposure these people had to actual particulate matter prior to their death, which is, you know, that's why I'm almost kind of shocked that this paper is in Nature, because there's so little data other than this very visible picture here. It's pretty sparse in the data department, in my opinion. There is an editor note where they're like, this paper's here because they show this really cool, you know, they make this observation that's pretty monumental. Like, up until this point, people weren't really looking at these lymph nodes, I guess, which is kind of crazy. Okay, let me just close something here real fast and we'll continue. I'm checking to see if, guys, you gotta let me know if my stupid ad manager happens, like runs an ad again. I'm sorry, I don't know why it's not alerting me before that. Okay. Our domestic product is so gross, right? Are these lymph nodes ectopic? No. So these, ectopic would mean that those lymph nodes have formed in response to a disease. So it's like they've formed somewhere where a lymph node normally wouldn't form, right? That's my definition of ectopic. These are lymph nodes that are from the lung. So they're like, they should be normal lymph nodes, if that makes sense. Okay. Oh, like a tertiary lymphoid structure, like TLS? Roslyn, are you an immunologist? So sometimes what happens, well, actually, there was this interesting research where, so people with brain tumors, obviously that's a really bad disease. It's really hard to treat brain tumors. So there were some people who had this idea, like, what if we can trigger the formation of lymph node structures in the brain so that you can have a nice array of like T cells that are getting activated by the dendritic cells and other cells that are in the brain next to the tumor that can then go and fight that tumor. Obviously, it's not great to have a lot of immune cells activated in your brain because that could cause inflammation and then death because of the pressure. Also, Country Logic, thank you for gifting a sub to Roslyn. I appreciate that. But Yeah, no, these are not tertiary lymphoid structures like that. They are just normal, your run-of-the-mill lung-associated lymph nodes. So going back to this, so then they were looking at, okay, let me see where I was, hang on. So they were looking at their donors. Right, because the lymphatic vessels that connect the lymph nodes and tissues, one might expect that black particulates would disperse across other types of tissue-draining lymph nodes. However, the fact that particulates became entrapped in the lung-associated lymph nodes suggested local effects of particulate matter on lung immunity. We found that the particulates were contained within a specific subset of macrophages that was located in the T cell zone of the lymph node and not within the follicles. So your lymph node, on the outside of the lymph node, like the outer barrier, you have all of your T cells, and then you have your the thing that they're calling the follicle. They're called B cell follicles, and this is where all your B cells hang out. And then they kind of form something called a germinal center, and like typically if you have a healthy lymph node, you'll always have this nice arrangement where you have this swath of T cells on the outside, a big, nice, beautiful B cell germinal center in the middle. And then what happens sometimes in disease is that if you like look at the lymph nodes from someone, or for example, like an animal with like a viral infection like SIV, which is the primate version of HIV, you'll actually see that their lymph nodes are totally messed up. They're usually smaller and they have fewer T cells because, again, the virus kills a lot of their CD4 T cells, and you get this like really messed up architecture of the lymph node. So one of the things that people always look for is like, how, like, is the architecture of the lymph node messed up or is it fine? Here, though, they're saying that the architecture is fine, but they noticed that particulates were being contained by macrophages that were located kind of on that outside zone of the T cell area, or the outside zone of the lymph node. Importantly, the particulate containing macrophages exhibited reduced activation, impaired production of pro-inflammatory cytokines. So these are kind of like, you could think of them as like little molecules that help wake up the rest of the immune response. And they also had significantly reduced phagocytic capacity. So this means that those, like again, I mentioned earlier that macrophages are really important for like engulfing foreign objects and bacteria and viruses, and that's called phagocytosis. And if they don't, they're saying here that these macrophages didn't have that ability. Now, I think what's really weird is that in this paper, they do not show any data for this claim. And I'm not sure if that's like located in a different paper, but I was like a little bit appalled that this was published without that data. They only show this like visual data here. So I feel like I'm missing something. Maybe it's in the additional information area. But anyways, I just thought that was weird because they didn't actually show that the macrophages were functioning worse. They just say it. Okay, so let's go down here. Right, so how did they interpret this? Just gonna breeze through this. Our study shows that the pollutants in our environment have a direct and detrimental effect on the human immune system. Lymph nodes filter impurities and coordinate the clearance of harmful antigens and pathogens, but over decades, the lymph nodes connected to the lungs can become clogged with particulates, and as a result, they're not able to carry out essential functions of host events and immune surveillance. Okay, that seems like a rather strong claim, but sure. Our study raises important questions concerning the mechanisms by which particulates are contained by specific macrophage subsets in the lymph nodes. Again, I'm like not sure. They don't show any flow data or anything to back that claim up. It is unknown whether these macrophages are resident in the lymph node or whether they are derived from lung macrophages that migrate to the associated lymph node. It will be interesting to assess whether targeting certain macrophage populations could facilitate the clearance of particulate matter. Okay, so I want to see what they have in additional information. Oh, wait, is this a different, hang on, this might be, is this more data? Yes. Okay. This was the summary paper. Okay. So the actual paper that this is based off of is this one right here. That makes so much more sense. We were reading kind of like the attenuated version. Their actual research article with all of the data showing that the macrophages function worse is located at this link. And it is open access, which is awesome. So you guys can read it if you really like that type of thing. I'm not going to go through it right now just because... That was the news and views. Yeah, that was the news and views article. Sorry, let me catch up again with this. Is it metals in the lymph node? So they actually, maybe they show it in this paper if those are metals, if they characterize what that particulate matter is, because in the attenuated version, they don't talk about what it is, what those particles actually are. Yeah, okay, so here's a nice, this figure is actually pretty easy to look at. We kind of already saw this. So it is carbon. Somehow they know that it's carbon particulates. Oh, this is a much higher quality, higher resolution image. So here on the top, we see the lung lymph nodes that are all blackened. And then on the bottom, we see the mesenteric or stomach associated lymph nodes that are all nice and beautiful, as they should be. These are their donor information. Okay, that's great. Let's scroll through this a little bit more. Okay, now they're doing some imaging. So now they're using these little antibodies. These are like things that recognize certain proteins, and they have a little fluorescent marker attached to them. And when you shine a laser on it, it'll light up and show you exactly where that protein is located in your tissue of interest. And they're looking for macrophages. So they're using these certain CD markers. We're not going to get into it. CD68, macrophage marker. CD8 is a T cell marker. And they're looking at, oh, and CD19 is the B cells. And actually you can really nicely see that there are these little B cell like hot spots in the hot pink. That's really cool. And then I think they're also looking at the particulate matter as well. So that's going to be the white stuff. And holy crap. In these old, so these are lymph nodes, I believe. And they're looking at 56-year-old and 73-year-old lymph nodes. And all that white snowy stuff, which actually I'm a little bit sussed out by this image. It looks like they had to turn up the exposure because it has this film over it. I'm like kind of confused. Why did they do that? Figure C, what does it say? Confocal images of human lymph nodes stained for CD68, the macrophage marker, to show black particulate matter accumulation in different macrophage subsets. Why did they do it? Oh, okay. I mean, it would be really hard to like dissociate this lymph node and then run it through a flow cytometer, which I think would give you much more concise information on a cell by cell basis, because you're like analyzing a single cell. Here they're like trying to look at the whole tissue, and it looks like they've, I don't know. I don't like how they did that image. That's fine, whatever. Sorry, let me go back to you guys. Did they compare? Did they compare smokers and non-smokers? Is that any data about the kind of lives? Is there any data about the kind of lives they live? Yes, I'm sure there is. I will find that, because I think they make it would obviously be very important if these people were all just smokers, right? Then that would make sense. Rosalind, you'll be working in a cancer immunology lab looking at exhaustion next academic year. Oh, that is exciting. Are you going to be looking at, well, obviously T-cell exhaustion, probably. That's super cool. I also work in a cancer immunology lab. Oh my gosh. Congrats on, I'm guessing you're going into grad school, it seems like. Congrats on that. That's big. Okay. I got to hold the first cancerous lymph node I had. It looked like a fleshy three-sided throwing star, had a black center and 3 weird triangles pointing out from the center. Whoa, wait, no way. If that's wild. That's really cool and horrific at the same time. They definitely mess with brightness and contrast. Yeah, I'm just like, why did you do this? You know what, like, obviously there's a difference, but I don't like that they didn't do that in their control. They did it in their control. Okay, it's fine. Although it looks slightly higher in their non-control, right? Like if you're looking at the white fuzzy film, it looks brighter in this image than in the control or the younger person. Yeah, let me see if they have if they talk about the people. Okay, here we go. Human samples. The lymph node tissues were obtained from brain dead organ donors through an approved protocol. Brain dead. So they were still alive when they took out the lymph nodes. Tissues for this study were obtained from donors with no history of asthma who were negative for SARS-CoV-2, cancer, Hep B and C, and HIV. We also selected for donors who were indicated in the donor information sheet as being non-smokers and or with no history of heavy smoking. Heavy smoking is defined as more than 20 packs for 1 year or more, which was indicated by 82 out of 84 donors. They have this in an extended table, which we're not going to look at, but if to your question about the donors, were they smokers? They were not. So they're trying to look at like specifically what happens to the lungs due to pollution, which again, now I'm like, do they talk about air pollution at all and like how much exposure these people had? That was the question I had. Maybe that's in the introduction again. Let's zoom back up there. Sorry, I'm like all over the place. Maybe that's what that first bit was up here. No.
Do you know the way?
Do they not talk about where? Their hypothesis was that inhaled atmospheric particulates and their accumulation with age may result in specific alterations to the immune function and architecture of the lung lymph nodes. I guess if you just kind of dissociate the study from like known exposure, if you just look at, because you can at least do the analysis where you're looking at, okay, this lung lymph node had way more particulate. Let's pair it with how well their macrophages function, for example. You could look at that and make a correlation there. Like if a lung has more of this carbon particulate in it, then the macrophage is less functional. But you can't say that, like what if these people all just came from areas where they were exposed to a lot of carbon particulate? And this is from New York. I think they said these were organ donors from New York. So this is like a big city, obviously. Yeah, these are from people that live in the New York metropolitan area. Okay, so these are people in a big city. So they're going to probably have a lot of exposure to like, you know, air pollution. I don't actually know how bad the air pollution situation is in New York, which would be interesting. But that's a good bit of information. We can kind of... I think the other information that's missing, though, is like how long were those people living in New York? Were they there their entire life? Were they there for a few years? You could imagine like that would greatly impact how much particulate is in these people's lymph nodes. But yeah, anyways, just some speculation and thoughts I'm having. Oh, it's a master's project. Oh, that's awesome. Do you plan, Rosalind, to go into industry after, or are you kind of like dipping your toe in academia to see if you like it? You used to live in New Jersey and there were big gas tanks from the gas refineries near homes. You lived near one. How long were you there, Nippon? That seems kind of bad because they would be giving off a lot of toxic things. Okay. Let me see. Do we want to look at any of this other data? A lot of this is imaging data, which is really easy to look at. I wanted to find what they claim to be functional data. Let's see. So now they're looking at disrupted lymph node architecture. Okay, so I was wrong. There actually is some disruption. They claim that Particulates disrupt lymphatic drainage. So this is again important for clearing the lymph node of the particulate. How do they measure drainage? It looks like they're measuring PDPN, which I forget, or podoplanin. Podoplanin? I forget what function that protein has. Oh, podoplanin, whatever this word is, you guys can see it here. It is a marker for the lymphatics. So these are the little like tubes where the lymph node cells can drain through. And they're saying that there's less of that in the people with particulates, it looks like. Interesting. Okay. I guess that's like the best you can do when you have a lymph node and you can't really do functional stuff with mice, for example. Okay. So here they're looking at these macrophages. Okay, now, okay, this is where they were talking about the altered cytokine production. So it looks like they're only looking at one type of cytokine, which is TNF alpha, but macrophages make a lot of TNF alpha, and TNF alpha is pretty important for sort of like triggering a strong immune response. It has good and bad effects, but usually it's indicative of how functional your macrophage is, if it's able to make this TNF alpha protein. But I don't like how they do this. They're literally just looking at the macrophage in green and then TNF alpha in red, and then they're kind of like overlaying it. I believe the overlay is the red, green, yellow image. It's really hard to see. I don't love this. You can see where the orange arrows are. Those are macrophages that are producing TNF alpha. The white snowy stuff, that is the particulate. So in a 39-year-old, they're like pointing to these little TNF alpha producing macrophages. They're like, ooh, look at all of these. They're pointing in the pink arrow to a macrophage that seems to have some of that white particulate matter in it. And they're pointing to, in the red frame on the right, you can see that there's no clear red spot, which would indicate TNF alpha being present. So they're saying this macrophage that's all big and containing the particulate, it's not producing the TNF alpha. Interesting. It's really, okay, the thing that I don't like about these types of images is that you can just kind of cherry pick where you pick your representative image in maybe an area where you have more or less macrophages making TNF alpha, and you can kind of pick that area. I don't know. It's kind of, I don't love this, but they do quantify. Here's the quantitative data where they actually take a whole bunch of these images and then quantify how much TNF is present. So the red dots are TNF alpha producing macrophages without black particulates, and then the blue squares are TNF alpha producing macrophages with the black particulate. And so the big takeaway is that the blue squares, especially in older adults, there's a smaller number of TNF alpha producing macrophages with black particulate than there are in the macrophages that don't have the black particulate, which suggests that something about the black particulate is making the macrophages unable to produce TNF alpha. Does that make sense? I feel like as I was saying that, I'm like kind of tripping over words in my head, but hopefully that kind of makes sense. If not, we'll move on. I didn't want to focus too much on this. I just kind of got distracted by the pretty pictures. You lived 13 years next to that? Oh gosh, that's intense, Nippon. You're leaning towards doing a PhD, but definitely want to feel it out first. I've done a bit of work on mitochondria, which was fun, but I love immunity, so I'll give that a go. Then exhaustion is like a perfect combination. I don't know if you've heard of this guy. Oh my gosh, what's his name again? Hang on. Let me think about it. Why am I forgetting his name? He's at University of Pittsburgh. He is a professor there that studies exhaustion in T cells. And I'm totally forgetting his name, but I wanted to, at some point, do my PhD with him. And actually, they accepted me, but I chose to go somewhere else. But he was really cool, and I'm forgetting his name. It starts with a G. His last name starts with a G. I think. God, what's his name? Delgoff. So the Delgoff lab, and what's his first name again? Peter? Was it Peter? Some super... where is it? Where is it? Greg Delgoff. Oh my gosh, I can't believe I forgot his name. So he studies T-cell exhaustion, and specifically with a focus on the mitochondria. I feel like that's like exactly your area of research, Rosalind. I don't know. But yeah, anyways, that was kind of an aside. Stromal cells, like the FRC, express PDPN, I think. Okay. The air quality in New Jersey is much better than it was even in the 90s. That's a relief for you, Ip. That's good. Great name for a sci-fi novel about a dystopian, disease-filled world. Black particulate, right? That would be great. I actually feel like this is slowly going to be more relevant when I decide to play Fallout a little bit later, because of like the dystopian world covered in particulate matter. Must be a Greg if you can't remember. Styles, are you a freaking mind reader? You're a freaking mind reader. It was Greg. I said a gas refinery, but I think it was actually a gas plant. Not sure if there's a difference. Gas refinery, I actually don't know if there is a difference. I feel like gas plants and gas refineries are similar. They're both burning off byproducts of petroleum, right? Do we know what the particulate matter actually is? That seems relevant. That is also another great question that I totally forgot about, but also was wondering, because I think I had mentioned earlier they claim it's carbon. But I don't know. Let me see. Let me just Command F. Like, how do they know it's carbon? See methods. Okay, let's see if they have this in methods. Accumulation of they keep saying the accumulation of carbon particulates. Pollution from carbon-based sources. Okay, this is not... Do they not say? What the particulate is? Oh, they did do flow. I guess I missed that. Oh, they did a phagocytosis assay. So they actually, yes, they actually did a functional assay. Oh, they used a macrophage line. Oh, okay, here we go. They did a phagocytosis assay. They used a immortalized human macrophage line called THP1 cells. These weren't from the human patients. This is like derived from a human a long time ago, and they've maintained these cells in the lab, and they can kind of live forever. They're not... I always get a little bit wary when you're studying macrophages, because when you do things to a macrophage to make it live forever, it's not going to be the same as the macrophage that you take out of a human body anymore. But anyways, it's the best proxy that we have. So here they're using that human macrophage line. Okay, good. They differentiated them using PMA. So this is to kind of activate them and get them to act more macrophagy and activated. And then they took atmospheric particulates purified from air filters and analyzed by the National Institute of Standards and Technology. There's a source for it, so maybe we can hop there in a second. Were purchased from Sigma Aldrich. So wait, apparently you can purchase purified atmospheric particulates collected from air filters. So they just got it in a little tube from this company to test the uptake of particulates. Oh, I see. The differentiated THP1 macrophage cells were incubated in fresh media with these black particulates. Following incubation, the cells were washed twice. Okay, blah, blah, blah. So they're using at at least in this functional assay, they're using these purified black particulates from air filters. I don't like that they have zero information about what that actually means. The source that they're putting here is called Development of Two Fine Particulate Matter Standard Reference Materials for the Determination of Organic and Inorganic Constituents. I feel like that's not going to answer our questions, so I'm not going to go there. Oh, I see your quote. Hang on. Oh, we're in an ad break. Shoot. I'm going to just go ahead and read Rosalind's quote, though. They said that black particulates are present in the atmosphere and consist of different polycyclic aromatic hydrocarbons derived from environmental pollutants such as motor vehicle exhausts, heating, and wildfires. Yes. But I just wonder, though, like, were they able to actually, like, isolate the particulates from their lymph node samples to see what those were? Because they could be different. I just feel like there's, yeah, I don't know, like, are all black carbon particulates the same thing? Are they all like these polycyclic aromatic hydrocarbons, or are they other things as well? I don't know. Maybe I'm just like overthinking this. But yeah. You'll check out, yeah, definitely check out Greg Delgoff if you're interested in mitochondria and T-cell exhaustion. He's kind of like the dude in the field, like, that does it, that focuses on that. And he's a nice person. Okay. So I'm sorry, I wasn't able to answer your question what the particulate matter that they took from these lymph nodes actually is, but as Rosalind pointed out in the paper, they are thinking that the black particulates are different types of polycyclic aromatic hydrocarbons. I'm guessing, like, usually an immunology lab too isn't equipped for analyzing particulate matter. Like, you have to send that off to a chemistry lab, and if you don't have that collaboration in place, then it's really difficult to actually do that. So maybe that would be a great follow-up for a future paper. And I'm actually curious, let's read their discussion section, because this is usually where they talk about, like, their future plans. So here, yeah, and they kind of recap what they did. So we show that particulates are contained within the T-cell zones, I believe, in the lung lymph nodes. These are all great acronyms. But they're not present in the T-cell zones in the mesenteric lymph nodes or stomach lymph nodes. Particulate-containing macrophages exhibit decreased phagocytosis over age, probably due to the direct effects of particulates, which I don't think they answer or, like, find out what those direct effects of particulates are. They just know that if the macrophage contains the particulate, it has decreased cytokine production, and this can exacerbate age-associated inflammation, which I'm actually not sure how they can make that link, but we'll just continue onwards. Moreover, the increased particulate content after the age of 40 results in disruption of the lymph node structure and lymphatic connections. Our findings provide evidence for individual and cumulative effects of environmental insults and senescent changes on lung localized immunity. Then they talk a little bit about the macrophages. So the macrophages that orchestrate the innate immune response through their phagocytic uptake of pathogens and the production of cytokines for immune cell recruitment and the initiation of adaptive immunity. So these are all important functions of macrophages. They also help maintain tissue homeostasis through uptake of dead cells and debris. So they're kind of like, we lovingly call macrophages the little garbage trucks of the body, because they're important for cleaning up, like, dead cells. Here we provide direct evidence that the macrophages take up atmospheric particulates, because that's debris, right, that lodge in the lung lymph nodes. Whether these macrophages originate in the lungs is not known, although the increased prevalence of these CD68 macrophages, I forgot what CD169 is a marker for. CD68 is a kind of like a mature macrophage marker. Hang on, let me double check this. There are over 300 different types of CD markers, and I forget what each one does all the time, so I have to check it. Okay, it looks like it's just the standard macrophage marker. Does it have any functional significance? Maybe these are tissue resident macrophages. I'm not entirely sure. But let's see. So they aren't sure whether these macrophages are coming from the lungs or if they were already present in the lymph nodes. So that's actually kind of hard to figure out. You would have to do, yeah, that's pretty hard to figure out where they came from. Our results also indicate that the fate of the particulate containing macrophage could differ with age. Macrophages in the lymph nodes of younger individuals exhibited higher turnover, so they were replaced by newer macrophages more than those from older individuals, which could facilitate particle clearance. So as the, yeah, but I don't know if they actually looked at that. Interesting. All right, let me see. I don't know. This is actually pretty long. Okay. Do they talk about future directions? Not really. Anyways, main point is that it's not great to have carbon particulates in your lungs. They mess with your macrophages and potentially your overall ability to respond to future infections and stuff like that. And again, if you are just popping by and you want to read this article, I'll put the link here. I'll also put it in my Discord for later so you guys can have that reference. It is open access, which is nice. There's too many CDs to remember. Yeah, no, seriously. Gosh. Yeah. And then it turns out, like, so I study another type of immune cell called natural killer cells, and they have their own set of like receptors and things that you have to remember, and it's just great. And everything sounds the same. Like, it's hard. Immunology is hard.